process of allopatric speciation. You'll recall that allopatric speciation occurs when a population becomes physically separated into geographically isolated subpopulations, either by a vicariance event, such as the uplifting of a mountain range, or when some individuals from a population disperse and colonize a new area. The basic idea behind allopatric speciation is that once gene flow is blocked by some physical barrier, then gene pools of the now isolated populations can begin to diverge due to natural selection or genetic drift. And at some point, these gene pools may be sufficiently divergent that individuals from the two populations can no longer mate and produce viable offspring. And it's at this point when we might say that two new different species have arisen from an original ancestor. Now, this account of allopatric speciation actually leaves an important question unanswered, and that is, what mechanisms are responsible for maintaining reproductive isolation once two populations have diverged sufficiently to be considered separate species? In other words, when they're no longer geographically isolated, what actually prevents them from mating? We're going to return to that question at the end of this lecture, but first I want to turn our attention to this other mode of speciation, sympatric speciation, in which new species arise without ever being geographically separated. The mechanisms responsible for sympatric speciation are harder to imagine than those responsible for allopatric speciation. Why? Well, remember, speciation ultimately is the outcome of reduced gene flow among subgroups in a population, resulting in isolation and divergence of separate gene pools. In allopatric speciation, gene flow is obviously reduced by geographic isolation. But how can a barrier to gene flow arise in the absence of physical separation? Well, there are a couple of conditions under which such barriers can and do arise. Now, one way that sympatric speciation occurs is as the result of an accident occurring during meiosis, which results in the formation of diploid gametes. You'll recall that meiosis normally produces haploid gametes, each having only one copy of each chromosome type. Sometimes, however, meiosis doesn't work right, and homologous chromosome pairs will fail to separate during the first meiotic division. And if that happens, you'll create some gametes that remain diploid, and of course other gametes that have no chromosomes at all. Now, if those diploid gametes fuse, the resulting zygote will have 4 chromosomes for each chromosome type. They are what we call tetraploid. In general, we refer to the condition of having more than the normal 2 copies of each chromosome as polyploidy, and polyploidy can arise on occasion because of errors during meiosis. Now, often when these kinds of chromosomal abnormalities occur, the resulting polyploid individuals just simply don't develop into normal adults. But sometimes they do. They survive, and they're able to produce their own gametes. If the gametes of these polyploid individuals, but remember that the gametes of these polyploid individuals will themselves have an abnormal number of chromosome copies. So, for example, a tetraploid individual will produce diploid gametes. Meiosis reduces the number of chromosomes by half, but if you start with 4 of each type and you reduce it by half, you'll end up with 2 of each type. Now, what does polyploidy have to do with sympatric speciation? The answer is that once a polyploid individual occurs in a population, it's no longer able to reproduce with normal diploid individuals. The reason for this, again, has to do with the cellular process of meiosis. Imagine that a tetraploid individual producing diploid gametes mates with a normal diploid individual producing haploid gametes. The resulting zygote would be triploid. That is, it would have 3 copies of each chromosome. The problem arises when this triploid individual itself goes to make gametes. During meiosis, there'll be no obvious way for the homologous pairs, or in this case, homologous triplets, to come together and separate correctly, because there's an odd number of copies of every chromosome. That beautiful choreography whereby homologous pairs of chromosomes get separated during the first meiotic division just gets messed up. There's a third wheel for every homologous pair. So most of the gametes that are formed will have dysfunctional odd sets of chromosomes, almost certainly rendering the triploid individual sterile. Thus, with a single occurrence of polyploidy, an organism can become completely and immediately reproductively isolated from the rest of its population and act as the founder of a new species. Now, you might see a problem here, namely that a polyploid individual can only mate successfully with another polyploid individual that has an identical chromosome number, because that's the only way that the resulting zygote and the individual formed from that will itself be able to successfully undergo meiosis. And there's two ways that this can happen. One way is that two polyploid individuals with the same number of chromosomes, let's say they're tetraploids, arise at the same time in the same place in the same population, which is possible but seems unlikely. The other way this could happen, though, is if a polyploid individual is able to mate with itself, to self-fertilize. Well, it's for this reason that speciation occurring for polyploidy is especially common in plants, because many species of plants can and do fertilize themselves. Thus, when a polyploid individual arises, its own gametes can fuse to form viable and fertile offspring with itself, immediately establishing a reproductively isolated population within the range of the parent species from which it arose. In fact, botanists estimate that about 70% of all species of flowering plants are polyploids, and thus have arisen through some form of sympatric speciation resulting from this chromosomal abnormality during meiosis. Now, another kind of polyploidy event that may lead to the formation of new species can occur when two species hybridize. Now, normally, interspecific hybrids are sterile, again, largely owing to the way that meiosis works. If gametes from two species happen to fuse, the combination of the two different haploid genomes will yield what is effectively still a haploid genome. There'll be a larger number of chromosome types, but none of them will be homologous to each other because they've come from two different species. So again, during gamete formation and meiosis, these chromosomes won't be able to sort themselves out and make normal gametes, and that's why hybrid individuals are often sterile. But if we have a hybrid individual and it suffers a meiotic error, such that gametes are formed containing all of the cell's chromosome, and if that meiotic error-produced gamete fuses with another similar gamete, then again we can effectively have a new diploid genome that has two copies of all of the chromosomes from the original two species. And as before, that individual, which is both a hybrid and now a diploid resulting from a meiotic error, is reproductively isolated from either of the two species from which it was derived. The evolution of modern wheat is actually an interesting example of a new species arising as the result of hybridization and meiotic errors. Actually, bread wheat is the result of two separate events in which hybridization and then meiotic errors produced polyploids. The first event occurred, it's thought, about 11,000 years ago when two species of wild grains hybridized. The hybrid was sterile, as I said, because it had essentially one larger haploid genome. But it's thought that a meiotic error doubled its chromosomes, allowing it to produce viable polyploid offspring through self-fertilization. The result in this case is a species of wheat called emmer wheat, which is still cultivated in many places in the Middle East. Then about 8,000 years ago, emmer wheat hybridized with yet another wild species, again producing a sterile hybrid, which again underwent a meiotic error, producing a fertile polyploid. The resulting polyploid is our modern bread wheat, which actually includes the genomes combined from three original wild species. Okay, we've seen how chromosome abnormalities arising from meiotic errors can lead to sympatric species, but are there other kinds of mechanisms other than these large chromosomal abnormalities that can limit gene flow between different subsets of individuals in the same population without them being geographically isolated? This question actually takes us back once again to the black-bellied seed crackers that we've mentioned a couple of times, and the question of whether the two morphs of that species might ever become one species. The answer in general to that question is that this will happen only if selection acts strongly enough to prevent gene flow between the one morph and the other morph, the large-beaked and the small-beaked individuals. Now, whether or not that's likely to occur, or even possible for the seed crackers, is not known. So I want to take a look at another example that's been a bit more completely described. This is the case of the soapberry bug. Now, this insect is named for the group of plants on which it feeds. It specializes on feeding on berries of a family of plants called the Sapindaceae, which includes the soapberry tree and a number of closely related species found in Florida mostly. Now, these insects use a kind of hard beak, what's essentially an elongated feeding apparatus, to feed on the berry in the following way. They actually pierce their beak to the center of the berry and then pierce the seeds that are found in the center and suck out the insides of the seeds. That's what the soapberry does. Now, there are several species of plants on which soapberry bugs feed, but they all have berries of approximately the same size. And the beaks of all of these soapberry bugs are approximately the same length. The length of the beak has become adapted so that it actually is the right length to get to the center of the soapberry berry that they're feeding on. But in about 1950, several new species of soapberry trees were introduced from the tropics to Florida to be used as ornamentals in gardens. And as often happens, especially in Florida, these species escaped from gardens and established themselves in the wild, and they now grow quite prolifically. And furthermore, some populations of soapberry bugs began feeding on these exotic species as well. Now, the important thing to know about these introduced new species of soapberry trees is that they all have much smaller sized berries than the native species. Very quickly, those populations of soapberry bugs that were feeding on the non-native, smaller berried plants began to exhibit shorter beak lengths as compared to the populations of bugs that continued to feed on the native species. Actually, this is demonstrated by looking at museum collections, just as was the case in the peppered moth that we talked about a few lectures ago. You can see a transformation in the sizes of the beaks of these soapberry bugs in museum collections of these insects, with collections made before 1950 largely having beak sizes all about the same. And then after 1950, you see the gradual emergence of different sizes of beaks, with some beaks being shorter and some beaks being longer. And the difference here being that the shorter beaked individuals are the ones that are feeding on the smaller berried plants. This pattern is a kind of disruptive selection then, just as is the case for the black-bellied seed crackers, with selection acting to produce soapberry bugs with two morphologies specialized for feeding on two different resources. So then how's it different? Might soapberry bugs represent an example of sympatric speciation in action? Well, it turns out that soapberry bugs actually mate and lay eggs on the same species of plants on which they feed. This is a very common phenomenon in insects. They develop an affinity for what we would call the host plant. So soapberry bugs that are feeding on one particular species of soapberries will prefer also to mate on that species and lay their eggs on that species. Now, the consequence of that is that the whole life history of soapberry bugs is tied to their host plant. And what this means is that there is an association between feeding preference and mating preference. Bugs are more likely to mate with other individuals they happen to encounter on their same host plant. And these individuals tend to have the same beak sizes because they're feeding on that plant as well. As a result, natural selection for beak size and the association of feeding with mate choice may eventually restrict gene flow, because large-beaked individuals and small-beaked individuals simply never run into each other. It's not that selection itself is acting to separate the gene pools of these populations. That's happening as a happenstance of the fact that selection is acting on the feeding specialization. So if the preferences for mating and feeding are strong enough, then over time we would imagine that selection on feeding will eventually isolate the gene pools of these two morphologies of the soapberry bugs, that they will become reproductively isolated because of selection and drift acting differently in the now isolated populations. Okay, those are a couple of ways in which sympatric speciation is thought to occur. And as I said, sympatric speciation is harder to demonstrate, less easy to understand, and is still actually quite a source of research in evolutionary biology. What I want to do now is to return to the question of what mechanisms are responsible for maintaining reproductive isolation once it's arisen. Remember that reproductive isolation between genetically isolated populations initially evolves simply as a byproduct of genetic divergence, that byproduct being either selection or drift in the case of geographically isolated individuals, or in the case of polyploids, the source of this genetic incompatibility between newly created species is really very obvious, having to do with the inability to simply sort chromosomes into gametes. Ultimately, new species will only result if genetic divergence does lead to some incompatibility that reduces mating and thus gene flow even after the initial isolating event or the initial isolating mechanism is somehow removed. There are two major categories, I should say, of ways that reproductive isolation occurs, or we might call these categories of reproductive isolating mechanisms. There are mechanisms that we refer to as prezygotic barriers. Prezygotic barriers are those factors that prevent the gametes of two individuals from ever possibly getting together. We call them prezygotic because they occur before the zygote is formed, before gametes have a chance to fuse. And there are a number of kinds of prezygotic mechanisms that we see that prevent gene flow between two reproductively isolated populations. These include, for example, a number of behavioral mechanisms. For example, many species use elaborate mating signals and displays that have to be precisely coordinated between two individuals before they'll ever copulate. Specificity in where mating occurs is the example of the soapberry bugs. This is also a kind of behavioral isolating mechanism. It's the behavioral preference for the host plant that will maintain isolation, genetic isolation between these two populations. Prezygotic reproductive isolation also can be due to mechanical, or we might call them morphological mechanisms. For example, in many species of insects, the shapes of genitalia are such that different species literally can't fit together to mate. Finally, reproductive isolation can be caused by physiological mechanisms. Basically, physiological conditions that are necessary for fertilization to occur may be incompatible between two species. For example, the sperm of one species may not be able to survive in the reproductive tract of another species long enough to reach the egg because of differences in pH or other aspects of the chemistry of the reproductive tract that simply causes that sperm itself to be non-viable. You can imagine there are any number of ways in which genetic divergence might give rise to such physiological incompatibilities or behavioral compatibilities or mechanical incompatibilities. In other cases, the gametes of two species may have the opportunity to form a zygote. But even in these cases, reproductive isolation may be maintained through what we call postzygotic barriers. That is, barriers that occur after gametes have fused to form a zygote. Postzygotic reproductive isolating barriers largely are caused by the fact that hybrid offspring of crossed species may not be viable. For example, the case of the inability to produce normal gametes because of the lack of homologous chromosomes. Or if they're viable, they may not be fertile. Or if they're fertile, they may be simply less vigorous and therefore less fit. Now, it's important to bear in mind that all of these reproductive isolating mechanisms maintain a barrier to gene flow between genetically divergent populations, but they're not themselves initially responsible for the formation of new species. Some other mechanism, such as geographic separation in the case of allopatric speciation, or chromosomal differences, or mating preferences for feeding specializations, as in the case of the sympatric speciation examples I gave, were initially responsible for restricting gene flow. Now, it's probably sometimes the case, maybe often the case, that populations that are initially isolated, reproductively isolated for some period of time, can come back together in contact at a point where they have diverged to some extent, but not sufficiently to prevent them from mating. And in these cases, we might expect that we would have the frequent hybridization of these two incipient species, but nonetheless, because of postzygotic mechanisms, the formation of offspring that simply weren't as healthy, simply weren't as vigorous as the offspring that would result from mating within species. At this point, you could imagine there might be strong selection to enhance existing reproductive isolating mechanisms. In other words, there would be a cost to individuals to hybridize, and so selection would act in such a way that traits of individuals would evolve to further prevent the possibility that they hybridize. The enhancement of reproductive isolating mechanisms by selection in this way is called reinforcement. This name derives from the idea that the traits under selection reinforce any existing reproductive isolating mechanisms that might have begun to arise when the populations were genetically isolated by some other means. The idea behind reinforcement makes a lot of sense because it seems fairly obvious that selection should be strong in these cases. But surprisingly, there's very little evidence to show that reinforcement occurs. Now, the best data supporting the idea of reinforcement actually comes from work on different species of fruit flies, different species of Drosophila. There are many species of Drosophila, and these species have different patterns of overlap in the wild, sometimes being allopatric, sometimes being sympatric, and also different patterns of the rate at which they'll hybridize. Jerry Coyne, who's at the University of Chicago, and Alan Orr at the University of Rochester did an interesting series of experiments a few years ago where they looked at the probability of any two species of Drosophila hybridizing and asked the following question. Are different species more or less likely to hybridize if they occur in sympatry or allopatry? What they did was they chose pairs of species that they knew from other evidence were about equally distantly related from each other. But in some cases, these species would occur normally in allopatry in different places, and in other cases, these species would occur in sympatry. The prediction is that if reinforcement is acting to enhance reproductive isolating mechanisms, then the species that occur normally in sympatry should be less likely to mate, even though they're equally genetically diverged from species that occur in allopatry, because the fact that they occur in sympatry means that there is an opportunity to hybridize and therefore an opportunity for selection to act to promote reproductive isolation. And indeed, when they did their experiments and asked which were more or less likely to hybridize, they did find that species that normally occurred in sympatry were less likely to hybridize, consistent with the idea that reinforcement does act to enhance reproductive isolating mechanisms. Whether or not that result is going to prove to be generally true of other kinds of wild populations of organisms remains to be seen. Okay, we've talked now about the process of speciation from a couple of perspectives. We've talked about it from the point of view of asking what species are. We've talked about it from the point of view of asking how new species can arise in different mechanisms. We've talked about it from the perspective of asking how species barriers are maintained by reproductive isolating mechanisms. Now what I want to do at the end of these lectures on speciation is turn to a couple of other final points that I think are important to talk about when we're talking about the origin of new species. The first one is this. Up to this point, we've been talking about the evolution of new species as though they were the result of the gradual accumulation of differences in genetic structure of populations that occur gradually over time. This view of evolution is called gradualism, and it's the one that Darwin espoused in his own writings. The view was that evolutionary change would be slow and steady, and the formation of species would be some slow and steady split, continual splitting of populations in two, so that if we looked at the evolutionary history of a group of species, we would find the gradual splitting off of a species here and a species there as those species diverged. More recently, biologists studying the evolutionary history of groups of organisms for which there's an extraordinarily rich fossil record, organisms such as clams and mollusks, where you can find just rich beds that extend deeply through evolutionary time and numbers of individuals to look and measure the morphology of. They've begun to argue that when they look in detail at the patterns they see in the fossil record, they don't see the gradual emergence of species, such as Darwin's gradualism proposed, but instead they observe a much more abrupt process. They find that, in fact, there's very little change in species for long periods of time in the evolutionary record of that species, and then all of a sudden, the very rapid and abrupt appearance of one or more species all at the same time that are quite distinct morphologically, even when they first appear. This alternative pattern of the evolution of species has been called punctuated equilibrium. The idea was proposed in 1972 by Niles Eldredge at Columbia and Stephen Jay Gould at Harvard. It's called punctuated equilibrium because the idea is that species normally are in equilibrium, relatively unchanging for vast periods of evolutionary history, which are periodically punctuated by very rapid appearance of new innovations. Now, what do we mean by rapid in this case? Well, for example, if we consider the lifespan of a successful species in some fossil record to be about 5 million years, what would be found is something like the majority of morphological divergence when new species appear occurring, say, within only the first 50,000 years. Now, 50,000 years seems like a pretty long time. Certainly many generations of organisms could appear in that time, but it's actually only about 1% of the total lifespan of the species. Indeed, Gould and Eldredge have pointed out that the isolation of small peripheral populations, as we've discussed might be the origin of species in allopatry, would be expected to cause very rapid divergences, especially because of genetic drift. The main point made by Gould and Eldredge was to argue that the process of speciation itself is what stimulates evolutionary change on the large scale. That once speciation has been completed, however, the evolutionary fate of most species is not to continued evolutionary change, as Darwin proposed, but instead evolutionary stasis through time. Now, the last point I want to address today is to ask the question of whether natural selection fashions perfect organisms. We often think, mistakenly, that evolution is somehow progressive, but it's not. Evolution just fashions organisms, but they're by no means perfect. And there are three simple reasons to offer as to why that should be the case. The first reason has to do with history. Each species, each population has its own historical legacy. And so evolution doesn't act on a blank slate, but instead has to modify and expand on the existing features that an organism possesses. In other words, the outcomes of possible evolution on an organism are constrained by prior evolutionary history. The second reason is that adaptations often are compromises. A particular anatomical structure or behavior or other feature of an organism may have very different competing selective pressures on it. For example, an organism might benefit by being brightly colored to attract mates, but the same bright coloration might also attract predators. So selection acting for mate choice would tend to make the animal brighter, but selection acting because of predation would make it duller. The result is a compromise. It's neither perfect for either function. And finally, we have to always remind ourselves that not all evolution is adaptive. Natural selection is an adaptive process, as Darwin first proposed, but other evolutionary mechanisms are equally or more important in changing the allele frequencies of population, most notably genetic drift, which is a random process. And so when drift is imposed on a population, it will change evolutionarily, but not in any sense that makes that population more adaptive or more perfect in any sense of the word. Next time, we're going to come back and look at other ways to uncover the evolutionary trajectory of species as a way to understand relationships among the multitude of organisms that exist on the planet, and a way to gain further insight into the significance of Darwin's view of evolution as descent with modification.